We're going to start the day with Dr. Jeanne Loring. She's going to talk about stem cells on Earth and in space.
Thank you, Danielle. Let me share my screen. I wanted to say what I did on International Women's Day was submit a grant, but also my 10-year-old niece came running in right in the middle of something I was doing, trying to kind of do on my computer. And she said, "Happy International Women's Day." And then she ran around and she came up and hugged me. She was so excited. Okay, so let me tell you about the work that we've been doing on the ISS. Okay, so I'm going to talk about work that we've done over the last, I guess it's been 4 years, a bit more than 4 years, with the use of stem cells for in microgravity. We're interested in knowing how cells behave in microgravity, but we're also interested in finding out whether there are things that can only be done in microgravity. And my focus is on stem cells. I am a professor emeritus at Scripps Research Institute. I am the senior science advisor for the National Stem Cell Foundation, which is sponsoring the work that we're doing. And I am also the founder of Aspen Neuroscience. Let me introduce you to stem cells kind of quickly. I think most of you probably have heard this, but let me clarify it. Stem cells are cells that can self-renew and can differentiate into 2 or more cell types. So there are 2 types of stem cells, and it's important to realize that these are very different from each other. Pluripotent stem cells can make every cell type in the body. They're pluripotent. The cells that can do this are called embryonic stem cells or induced pluripotent stem cells. The other stem cell type are the tissue or adult stem cells that are present in our bodies. They can only make a couple of cell types, generally just the cell types in the organ in which they're found. And there are 2 general types. There are mesenchymal stem cells, which come from connective tissue, and there are fetal and amniotic stem cells, also associated with cord blood, birth-associated stem cells. But all of these can only make a couple of cell types at most. Oops, this way. So let me tell you about pluripotent stem cells. The reason they're so exciting is that they can differentiate every cell type in the body. They are not in our bodies now as adults. You did, however, have them right after you were a fertilized egg for about 5 days after fertilization. These cells exist and they can be captured. But after that, you no longer have any pluripotent stem cells in your bodies. This is an illustration I like to use of what all pluripotent stem cells can become. So imagine the power of having cells in a dish that you can make into every cell type in the body. The adult stem cells are what are called multipotent. That means they can regenerate the tissues in which they're located. And I've shown that they're just little parts of this pluripotency tree. The main types are bone marrow and also umbilical cord blood cells, which are hematopoietic stem cells. These are used for cancer treatment. They're also being used for other gene therapies in which you can deliver a corrected gene via hematopoietic stem cells. Hematopoietic stem cells are approved by the FDA. And however, pluripotent stem cells and also connective tissue cells have very limited approval from the FDA. Connective tissue cells, which are often known as mesenchymal stem cells, can come from all sorts of parts in the body. I'm not going to talk about the clinics that are unapproved that are offering stem cell therapies, but in general, they derive their mesenchymal stem cells or cells like those from adipose tissue, from liposuction. And that is not approved by the FDA. And my personal opinion as a scientist is that they don't do anything. The sources of pluripotent stem cells are, first, in 1998, they were made from discarded embryos that were left over from in vitro fertilization procedures. This is a blastocyst stage, which is the stage at which embryos that are fertilized in vitro are normally transplanted to the uterus. The inner cell mass, which is this small little group of cells, there's only a few hundred of them, but they are the cells that will give rise to the entire person, the entire fetus. The inner cell mass can be captured and cultured, and you can create lines of embryonic stem cells, which continue to self-renew and continue to have the ability to make every cell type in the body. So there's another type of pluripotent stem cells, which has proved, I think, to be much more transformative in the stem cell field. And these are cells that are derived from skin biopsies or from other cell types from volunteers. If you use a skin biopsy, take the fibroblasts from the skin, grow them up in a culture dish, and then add to them reprogramming factors, which are transcription factors that temporarily reset the entire genome. So the genome now has the same kind of activity as the embryonic stem cells have. These cells are also able to give rise to every cell type in the body. And I think it's important to note that these cells can come from anybody and match you, unlike embryonic stem cells. This is so important that, and so remarkable, that the Nobel Prize was awarded to Shinya Yamanaka in 2012 for his discovery in 2007. So stem cells on Earth, what can they be used for? They're used for understanding genetic diseases because they carry with them. You can get the cells from people with genetic diseases. You can know everything about their disease. And if it's a known disease-causing mutation, you might see a manifestation of that cell, of that mutation in cells that you can make in the culture dish from iPS cells. So a couple of examples are from my own lab are Friedreich's ataxia and Fragile X syndrome. These are both neurological diseases, and so the cell type we made from the iPS cells, or the cell types we made from the iPS cells, are neurons, central nervous system neurons. They are also in drug companies being used for preclinical drug screening for both safety and efficacy, especially, for example, if you make neurons, you can use those for neurotoxicity studies. And also in my lab, we have made ethnically diverse populations of iPS cells in order to discover differential responses to drugs based on genetic background. Cell-based therapies are what usually you hear about in the news. The idea is to use cells for delivery of therapeutics or for replacing cells lost to disease. Using the cells for delivery, a couple of examples are one quite remarkable discovery we made several years ago, in which we put enzymes into stem cells and then put them into the brain in a mouse model of Alzheimer's disease, or a mouse model that looks a little like Alzheimer's disease. And we discovered that by delivering that enzyme, it wasn't the cells, but delivery of the enzyme, degraded the amyloid plaques in the brain. Another example that from my lab is delivery of anti-inflammatory factors to multiple sclerosis, in which we put the cells into the spinal cords of mice that had been induced to have the symptoms of multiple sclerosis, and they recovered. Right now, probably, and certainly my greatest focus is replacing cells lost to disease. And the example from my lab is Parkinson's disease. I'm going to briefly tell you about that. So there are a number of cell replacement therapies using pluripotent stem cells that are now in development or even in the clinic. One of them is natural killer cells, or CAR T cells. These are used to treat cancers, and this is in clinical trial stage. Retinal pigment epithelium can be made from iPS cells to treat age-related macular degeneration. There's also a clinical trial in progress for that. Neurons and or glia have a lot of applications, including Parkinson's disease, Huntington's disease, spinal cord injuries. And those are either in clinical trials or in preclinical, so they are working their way towards the clinic. Epidermal cells are being examined for their ability to restore hair loss. That's in preclinical stage. Cardiac cells for heart disorders, pancreatic beta cells for type 1 diabetes. The type 1 diabetes work is already in clinical trials and has been quite successful in some cases. So that's the future right now, and the future is right around the corner. The work that we're doing is on Parkinson's disease. And the reason why stem cell therapy makes sense is because Parkinson's disease is caused by the loss of a particular neuron cell type, dopamine neurons, in a particular part of the brain called the substantia nigra. This results in movement symptoms like tremor, rigidity, loss of balance, and they're all due because to the fact that there isn't enough dopamine input to the neurons in this part of the brain, which is called the striatum. So here's the pathway that we want to restore, and we're going to restore it, as you'll see, by placing new dopamine neurons straight into the target. So there are about 50% of dopamine neurons left in the substantia nigra when symptoms appear. And the drug therapy that most people are taking, L-DOPA, has only one way to work. It doesn't slow the disease. It increases the production of dopamine, which improves the effectiveness of the remaining dopamine neurons. But the neurons continue to die. It is not a curable disease at present. Cell replacement therapy for Parkinson's disease is the subject of my work at Scripps Research Institute over the last few years, and was the basis for my founding of Aspen Neuroscience, which is a company here in San Diego area. The strategy is to make iPS cells from a person with Parkinson's disease, then turn these cells into early stage dopamine neurons. These dopamine neurons are then transplanted to the brain, as illustrated here on the right, into the striatum, the target of those cells. And then if our animal studies are indicative of the result, including primate studies, by the way, these studies show that the transplanted human cells will grow out and reconnect with the host brain, restoring the function. So essentially restoring this lost connection by shortening it and putting the cells directly into the striatum. So I'm going to talk now, because I don't have a lot of time, about stem cells in space. Stem cell research that we've been doing on the International Space Station. And Elon Musk was correct when he said it looks hard and it's harder than it looks. So it took a team. This is a collaboration between the New York Stem Cell Foundation, the National Stem Cell Foundation, and Space Tango. And these people, Paula and Jeanne from the National Stem Cell Foundation, are the driving force behind the funding for this project. And this group of scientists, Valentina, Davide, and Scott, are at NICEF, and they do the bulk of the culture work and the preparation work. Then Twyman Clements, Danielle, and are the representatives from Space Tango, who have been responsible for essentially putting the product together so that it can be sent to the space station. The project that we've decided to do is one of hundreds that one can imagine. Since the cells are pluripotent, you should be able to study any cell type in microgravity simply by making it from iPS cells and sending it up to the space station. Our project is studying the effects of microgravity on neuroinflammation. The reason this is important is that neuroinflammation underlies every neurological disease, including Parkinson's disease, multiple sclerosis, and a number of other neurodegenerative diseases and neurodevelopmental diseases. The immune system in the brain is not the same as the immune system in the periphery. It's isolated from it's isolated from the blood system by the blood-brain barrier. So it has its own intrinsic immune system. And this consists of cells called microglia and astrocytes. The microglia are like the macrophages in the blood. And what I mean by that is that they are attracted to cellular damage and they engulf cellular debris. The cells that die, they essentially clean up everything. But they also are attracted to the places where there is damage in the brain. So they have a high probability of being useful for cell therapy, ultimately. Microglia can heal, but they can also damage the brain because they release what are called pro-inflammatory cytokines, which could lead to more neuronal damage and death. So we wanted to study this phenomenon in a culture system, an iPS cell culture system in microgravity on the ISS. And what we came up with was partly because if any of you have grown cells in a culture dish, you know gravity is kind of important for keeping the cells attached to the dish. They'll just float away. So, and also because we wanted to have something more representable, more representative of the brain itself, we mixed neural cells and glial cells into structures, three-dimensional structures that we call organoids. And we chose two diseases to study. We got iPS cells from a person with progressive multiple sclerosis and a person with Parkinson's disease. Now, we don't know whether there is a genetic basis for either one of these diseases in this particular case. So we're not sure whether they'll be different or not, but we did look at two different cell types. For Parkinson's disease, we took fibroblasts, made them into iPS cells, and then made them into dopamine neurons in the same way that I just described for the cell therapy. And we separately made microglia from those cells and then mixed them together. And they form these adorable little balls that contain both neurons and microglia, and they continue to develop. And we do this for both healthy individuals and for Parkinson's disease patients to see whether it makes any difference where the cells, the disease state of the individual from whom we got the cells. For multiple sclerosis, we did the same thing, but instead of making dopamine neurons, we made cortical neurons, which are the neurons in the frontal cortex. And they're responsible for a lot of brain functions. There are neurons that are affected in multiple sclerosis. So we did the same thing and made cortical organoids from both a healthy individual and a fibroblast. And by the way, these slides come from Valentina Davide and Scott at New York Stem Cell Foundation. And then we sent the cells on a long month's journey from the lab. They started out with a road trip. They were in the lab. We made the organoids, and then they were shipped to Cape Canaveral, in this case, Cape Kennedy, and the Kennedy Space Station. And they went on a truck, and then they were placed on a rocket, sent up to the International Space Station. And then they came back under the way that all things are recovered from the space station. They were they were dropped. They were sent off the space station and were splashed down in the ocean. So they also had a trip on a ship, and then they were sent again on a road trip, and then eventually ended up in the lab. So this is just to show you that, as Elon Musk said, it's not easy. We had to put all of these individual bits in place, and so it required the cooperation of a very large number of people. I'm gonna wind up now by telling you about just a really quick overview of what we're analyzing in these cells, what we're looking for. In every case, we have a ground experiment and a LEO, or low Earth orbit experiment. And we study the viability of those cells after they come back, the structure of the organoids. We measure their gene expression. We look at proteins that are secreted by the organoids, and we are planning to look at the genome integrity, discover whether their time and space had any effect on their genome. And we ultimately want to do electron microscopy and electrophysiology and probably anything else we can think of, because these samples are so precious. We want to know everything we can about them. Here are some of the things that we've done so far. This is the viability and histology. Over here, you can see that the cells that are in low Earth orbit this is an organoid that's been on the ground. You can see they get bigger in space, and they've been cultured for the same length of time. So there's something going on, maybe like loosening because of the effects of gravity, loosening of the connections between cells that make them expand in space. We don't yet know whether there's more cell proliferation. That was certainly a possibility, but that seems like it's unlikely to be the major contributor to the increase in size. When we brought the organoids back from space, we put them into culture dishes, and as you can see, they thrived. They grew lots of axons out into the periphery. They were perfectly happy and ready to make more synapses with whatever they could find. The histology showed that, showed that there were neurons surviving, and that they would form in the cortical organoids. They would form these little things called rosettes, which are a sign of developing cortical neurons. We also did gene expression analysis, and I'm not going to tell you anything about this because this is a subject of a paper we're writing right now. But you can see that the dopaminergic cells and the cortical cells were different from each other, and also different between the patient and the control. So this is where we're going to get a lot of information about individual variations, and perhaps some information about how the disease state affects the gene expression and the formation and growth of those organoids in space. So to summarize, we've had 3 missions. The most of our work has been from this one, which was, of course, studied in 2020 and then not studied for a while because of COVID. We now have all the results from that, and we are in the process of analyzing the results from this one, which was in 2021. And essentially what we've discovered is that iPSC-derived neural glial organoids survive and they thrive for 30 days at least on the International Space Station. In a sense, this allows us to have a window into the brain of astronauts. I'm not going to tell you that these are actually like the brain, but they have components of the brain. We found differences in gene expression and secreted proteins between the ground and space. And the most important thing is the manuscript we are now working on from Davide and the colleagues at New York Stem Cell Foundation, which we hope to submit very soon. These are the mission patches from the 3 missions. And I want to close by thanking the team. This is Paolo Grisanti, Valentina Fossetti, me, and Jeanne. And these are the other people, many of which were involved. This is not an easy thing to do, and we could not have done it without every single person who was involved. Thank you.
Thank you. That was fantastic and a perfect overview. I know I'm excited for your manuscript.
Me too.
I can only imagine.
No, it's great. I mean, actually, the data are fantastic, even though I didn't actually show them to you because I'm not ready to make conclusions until we've thought about it a lot more deeply.
Absolutely. And we have just a few minutes before our next session. So I wanted to ask one of the questions that we do have through the Q&As. Can you summarize the key advantages that benefit stem cell research in microgravity? Why microgravity?
Well, so my idea, I'm going to tell you what I think, and then I'm going to tell you more generally. I think that we need to find out about the health of astronauts in more ways than taking samples of their blood, which has been done extensively. You can't take samples of an astronaut's brain while they're in space. And so this could be a sort of brain avatar for astronauts so that you could find out whether that particular person might have a reaction, like a neuroinflammatory reaction that would compromise their health. More generally and speculatively, there are a lot of opportunities for screening drugs in space with stem cells. And that's probably a much more commercially applicable approach. We really don't know whether it's going to be better or not, but we suspect that both drug synthesis and drug screening will be different in space and therefore perhaps more helpful. We certainly need a lot of help. Both of those areas are very difficult on Earth.
Okay, thank you so much. This was a fantastic keynote presentation to start the day.
Thank you very much for inviting me, and happy International Women's Day.
Yes, absolutely. Happy
This
Women's
could
History
not
Month.
have been done without women, believe me.
Thank
Take care.
you. Thank you.
Bye, everybody.